Can somebody confirm you can hear me?
We can hear you.
Thank you. All right, before we begin, are there any questions? Not hearing anything, so let's begin. Let me turn on my screen. All right, there you should be seeing my screen. Let me change that. I don't have it optimized for, there's that. No idea how you switch, so I'm just going to get out of it. And there we go. Now it's optimized for video. All right, if there's no questions, let's begin. This is where we were last time. We go to the syllabus. So what we're supposed to be doing is Chapter 8, Microbial Genetics today. In the lab later today, we'll talk about Lab 9. Any questions about what we're doing? We should finish Chapter 8 today. I'm not sure we're going to make it. We'll see. Where are we? There it is. So we had talked about protein translation last time and mentioned that translation will stop when the ribosome reaches the stop codon, in which case the stop codon will fall apart, falling off the messenger RNA, and that will release the newly replicated protein that's been recently translated. Let me talk about some of the complications of translation, and that is when the protein is being translated, there can be more than one ribosome on the messenger RNA, as shown here, in which case we can have more than one protein being translated on the same messenger RNA. That's one complication that both eukaryotes and prokaryotes have, and that is the one messenger RNA can have more than one ribosome on it, making more than one protein from each messenger RNA. And as you can see, this one has two ribosomes on it, and as you that's actually the messenger RNA, they can have more than one protein being made from the same RNA. In prokaryotes, we have another complication, and that's shown in this picture here, and sort of in that picture there, and that is protein translation can begin before RNA transcription ends. And that's because RNA transcription is happening in the cytoplasm where protein translation happens. This doesn't happen in eukaryotes because in eukaryotes, transcription happens in the nucleus and then translation happens in the cytoplasm. But in prokaryotes, you can have the RNA being transcribed, and before the RNA is finished transcribed, uh, ribosomes can begin binding to it and then starting protein translation before the RNA transcript is even finished transcribing, meaning before the RNA is finished. And that's shown here where this is near the end of RNA transcription, and there we have a very long RNA, and you can see that it has multiple ribosomes bound to it, beginning to transcribe the RNA molecule even before it is finished. And this is a shorter RNA molecule, and it has multiple ribosomes bound to it. Shorter RNA molecule, shorter RNA molecule, shorter RNA molecule, and these are the two shortest RNA molecules, and even they have ribosomes bound to them. That one has two ribosomes bound to it. That one looks like it's like three or four. And the protein translation is starting even when the RNA molecule is very small, so that the RNA molecule has a large amount of RNA transcription to occur before the RNA is finished transcribing. Any question about that complication? That is only seen in prokaryotes. In eukaryotes, you don't have that, like I said, because RNA transcription happens in the nucleus, and then protein translation happens in the cytoplasm. But you still have in eukaryotes, you do have multiple ribosomes binding to the same RNA molecule. So you can have multiple proteins from the same messenger RNA. In eukaryotes, we have another complication, and that is in eukaryotes, you have a DNA molecule which will be transcribed, so this is a gene, but there's only portions of the gene, the exons, which are actually translated into protein or the final RNA product. You have portions of the gene introns which are initially made in the messenger RNA. The original RNA transcript does include the exons, the portion of the RNA that'll code for the protein, but the initial RNA has an intron which does not initially code for the protein. Those introns have to be spliced out before the final messenger RNA will be made. Spliced out means cut out, and then the exons will be spliced together, put together, to make the final messenger RNA. Curiously, only the final messenger RNA can leave the nuclear pore of the nucleus. That there's a checker on this pore, and if there's introns in that messenger RNA, it will not be allowed to leave the nucleus. So those introns have to be spliced out, and then the exons spliced together before that messenger RNA can actually leave the nucleus. And that's a complication, RNA processing in eukaryotes. This happens in eukaryotes because even in the gene, there are regions of the gene that do not code for the final RNA product, which is usually a protein, and those we call introns. Any question about any of that? With bacteria, we do not have introns in the gene. We only have exons in the gene, so you don't have this complication in a bacteria. All right, we talked about the structure and function of the genetic material. We talked about the flow of genetic information. We talked about DNA replication, RNA transcription, and protein translation. We're now going to turn to the regulation of bacterial gene expression, talking about that with an operon. We'll briefly talk about eukaryote gene expression, but mostly bacterial gene expression. Then after that topic, we'll talk about mutation, and then talk about genetic transfer and recombination. And lastly, we'll talk about genes and evolution. The regulation of a bacterial gene expression means that the cell can regulate what is in the cell. You can control gene expression, turning on a gene so that its products will be made, or you can turn the gene off, in which case the gene's product will not be made. There are different genes. There are constitutive enzymes, which are constitutive genes, and they're expressed at a fixed rate. There are other enzymes that are expressed only as they are needed, and the gene will be turned on when it is needed and turned off when it is not. There's two ways to do that, the repressible and inducible enzymes. We'll only talk about the inducible genes with an inducible operon. This is a pre-transcriptional control mechanism where the cell regulates what's in it by turning on the gene when it's needed and turning off the gene when it is not. We're only going to talk about the inducible operons. There are repressible operons. We're not going to talk about it, and we will not be tested on it. There are other means to control the regulation by pre-transcriptional control. There's an epigenetic control, and we're not going to be talking about it, and you're not going to be tested on it, and you will actually thank me for not testing you on epigenetic controls, because even today, geneticists do not have a good understanding and what do you say? Understanding of epigenetic control, meaning they have some understanding, but they kind of wave their arm and say, what's going on with epigenetics, and we're just not going to talk about it. There are also post-transcriptional control, so like you can make the gene product and you could either degrade the messenger RNA or inactivate the messenger RNA or inactivate the protein or degrade the protein. These are all post-transcriptional control. We're not going to be talking about it, and you will not be tested on it. Does it make sense for a gene, for a cell to make a gene if the gene product is not needed by the cell? Can anyone answer that question? It's either a yes or no question. Does it make sense for a cell to make a gene product if the gene product is not needed by the cell?
No.
Yeah, it doesn't make any sense for the cell to make a gene product if the gene product is not needed. And we can turn the gene off if the gene product is not needed. And that's what cells do. All right. So in talking, for some reason, my vision has gotten blurry here. Let me wipe these down. It's almost like the light is reflecting off my computer. You can talk about turning on and off the genes by using the operon model of gene expression. Bacterial genes use the operon model, and we're only going to be talking, well, we'll talk briefly about eukaryotic operons, but mostly we're going to be talking about bacterial operons. A bacterial operon does contain the gene of interest, and that we call a structural gene, shown here. So like when normally we're talking about the blue eye gene or the gene that codes for um, lactose degradation, meaning the gene for lactase, we're talking about a structural gene. Any questions about that? In a bacterial operon, you can have more than one structural gene in the operon. For example, for the lac operon, we have 3 structural genes in the lac operon. The Z gene, which is a structural gene, the Y gene, and the A gene. The Z gene is the gene that codes for the enzyme lactase, which digests the sugar lactose. The Y gene calls for the protein transporter, which is on the cell membrane, which transports the sugar lactose into the cell. The A gene is a gene involved in converting beta galactose to the sugar glucose. When you digest lactose, you make beta galactose and glucose. Glucose, of course, can be used easily by the cell, because it's the base sugar. But beta galactose has to be converted into glucose. And the A gene is a gene involved in helping convert uh, beta galactose to glucose. And that's all you need to know about the A gene. From this point on, I don't think we'll be talking much about the A gene. So those are three structural genes in the lac operon. The lac operon also has a promoter, which is a region of the gene upstream of the gene, which is important in getting the gene product, the lactase enzyme made. And you already know what the promoter is because we talked about it already. The promoter is where the RNA polymerase binds to the DNA. And then there's the operator, which is a region also upstream of the structural gene, which controls well, it turns on or turns off the structural gene, turns on or turns off the operator in reality. Any question about any of that? All right, so that's the operon. It does include the terminator at the end of the gene, or in this case, the end of the A gene, which codes for the RNA polymerase to fall off the DNA. And that's not shown here. What is not part of the operon is the I gene, which is a regulatory gene. And the regulatory gene controls the expression of the lac operon, meaning the regulatory gene determines whether the lac operon is turned on or turned off. And the regulatory gene is its own operon, so it has its own operon, which is not shown here. Any question about any of that? All right. As I mentioned, you can control gene expression either by induction or repression. We're only going to be talking about induction. We will not be talking about repression. Induction is a case of the lac operon and is inducible operon, meaning induction occurs in the lac operon. Induction means that the gene is normally turned off, but when the gene is needed, it can be turned on, meaning it will be turned on when gene is needed. Repression is the opposite. The repressible operon is an operon which is normally turned on, but the gene can be repressed or turned off when it is not needed. And we will not talk further about repression. Any question about that? So let's talk about the lac operon. It's an inducible operon. It normally is turned off. How it is turned off is the I gene, which regulates the control of the lac operon, turns off the lac operon, meaning it turns off the lactase gene. Normally, the gene that regulates another gene is not anywhere near the gene it regulates. In eukaryotes, normally the gene that regulates a gene in the eukaryote cell is usually on another chromosome than the gene it's regulating. However, in the case of the lac operon, the gene that regulates the lac operon is right next door to the lac operon. And this gene is called the I gene. When the I gene is expressed, it will make a repressor RNA, which will make a repressor protein. What that repressor protein does is it binds to the operator of the lac operon. And now when the RNA polymerase binds to the promoter and starts moving down the DNA, it will get stuck in the operator. And that way the Z, the Y, and the A gene will not be transcribed. Remember, the Z gene codes for the enzyme lactase. And that is how the lac operon is normally shut off. The repressor protein is made, and then it binds to the operator, and then the polymerase cannot get past the operator when the repressor protein is bound to it. Any questions about any of that? Well, there has to be a way of turning on the lac operon, and what we have is an inducer which binds to the repressor protein and inactivates the repressor protein. It turns out that allolactose is the inducer. Allolactose is an isomer of the sugar lactose. One is left-handed, one is right-handed. I think lactose is left-handed and allolactose is right-handed, but I could be wrong on that, so don't quote me on that. And you don't really need to know. All you need to know is allolactose is an isomer of lactose. And that means whenever the sugar lactose is around, allolactose is around. Allolactose will get into the cell and then bind to the repressor protein and inactivate it. Once inactivated, the repressor protein cannot bind to the operator. That will turn on the lac operon. And that means when the RNA polymerase binds to the promoter, it will go past the operator and transcribe the Z, the Y, and the A gene. Any question about any of that? Well, here's a question for you. Why does E. coli normally want to turn off the lac operon? Why does it want to turn off the lactase gene? Anyone have a guess? Well, where do you find the sugar lactose? Can anyone answer that?
Milk.
Milk and dairy products. How often is E. coli going to see the sugar lactose? Assuming E. coli is in the environment and not in somebody's gut who happens to be drinking milk. How often is E. coli, like on an apple, going to see the sugar lactose?
Never.
Almost never. So that's why the lac operon is normally turned off. But it does need to have a way of turning it on. And how it turns it on is whenever allolactose is around, and allolactose will be around whenever the sugar lactose is around, it will bind to the repressor protein and inactivate it. And that's how the lac operon gets turned on in a simple nutshell. Let's watch a little video on the lac operon, and then I'll talk more about the lac operon. Well, that didn't seem to work. Let me try that again. Apologies, I don't have the button here to expand this. I'm not sure why that's the case. Oh, there it is.
The E. coli lac operon is an example of an inducible set of genes. These genes are responsible for the breakdown of lactose into sugars used for cellular metabolism. This inducible system also involves bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. This animation will only focus on 2 of the 3 proteins encoded by the lac operon: beta-galactosidase and permease.
Let me say beta-galactosidase is another name for the enzyme lactase, and permease is another name for the protein transporter, which brings the sugar lactose into the cell membrane or into the cell. Any question about that?
Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose is the lac operon inducer molecule. After first appearing in the cellular environment, lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region.
Let me state that that is a little bit of an oversimplification. The lactose doesn't actually bind to the repressor when the repressor is bound to the DNA. It has to be a free repressor. And when it binds to the free repressor, it inactivates the repressor molecule. The lactose doesn't really bind to the repressor molecule when it's bound to the DNA. And this video is just a little oversimplified.
Cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated. The first protein is beta-galactosidase, which breaks down lactose into 2 simple sugars. The second protein is permease, a membrane-bound protein. When embedded in the cell membrane, permease functions to provide a direct route for the lactose outside the cell to be imported into the cell. This import occurs at a much greater rate than the passive transfer we first observed. Because translation continues inside the cell, other permease proteins become embedded in the membrane. This further increases the rate at which lactose enters the cell. Beta-galactosidase breaks the cellular lactose into the simple sugars glucose and galactose. Once its concentration is greatly reduced, the lactose bound to the repressor are released. At this point, the repressor again binds to the controlling region and gene expression is halted. For all inducible systems like the lac operon, it is the interaction of the repressor and inducer molecules that mediate gene expression.
All right, any questions about that video? So, normally the gene is turned off. Come on. Because the repressor gene is transcribed into the repressor molecule, and then the repressor molecule binds to the operator, and that turns off the lac operon, meaning none of these genes are expressed when the repressor molecule is on the operator. But if allolactose is around, and it'll be around whenever the sugar lactose is around, it will bind to the repressor molecule. And when it binds to the repressor molecule, it inactivates it, and now the repressor molecule cannot bind to the operator. That will mean when the promoter binds to the, when the RNA polymerase binds to the promoter, it will move down and then transcribe the genes. What do you notice which is different about this transcription that we've never seen before? At least I've never talked about it in the class before. How many proteins are being made from this messenger RNA? People, how many proteins are being made from this messenger RNA? One, two,
3.
yes, 3. Thank you. We've never seen that before. This happens in prokaryotes where the operon has more than one structural gene in the operon. There can be more than one protein made from one messenger RNA. You never see this in eukaryotes because eukaryotes only have one structural gene in their operon. But prokaryotes have related genes in their operon. I never mentioned that. In the operon, if there's more than one structural gene in the operon, they will be related genes. And in lac operon, you can understand the relationship. They're all involved in the metabolism of the sugar lactose. In eukaryotes, we do have a gene for lactase. We also have a gene for the protein permease, which allows the lactose to move through the cell membrane. And we have the gene A, which is involved in converting beta galactose to glucose. We have all three of these genes, but they're in three different operons because in the eukaryote operon, we only have one structural gene in each operon. In bacteria, we can have more than one structural gene in the operon. You don't have to have more than one structural gene, meaning there can be a bacteria operon which only has one structural gene in it, but there can be more than one structural gene in the operon. We're only going to talk about the lac operon, which has three structural genes in the operon. And this messenger RNA will code for three different proteins. Any question about any of that? All right. Well, here's a question, and that is, although the allolactose can bind to this repressor protein and inactivate it, what happens to the repressor protein that is already bound to the operator? I already mentioned that allolactose does not bind to it because the protein has to have the correct three-dimensional shape, and when it's bound to the DNA, it will not be in the correct three-dimensional shape for allolactose to bind to it. So does it always stay there? You already know the answer to that. It can't always stay there because the gene is sometimes turned on and sometimes turned off. So what happens is this repressor protein binding to the operator, this bond is a stoichiomic reaction, and every once in a while, the repressor protein falls off. If there's other repressor protein around, it will bind to the operator, and then this lac operon will continue to be turned off. However, if allolactose is around and this repressor protein falls off the operator, there isn't other repressor protein around to bind to the operator. And furthermore, allolactose will bind to the repressor protein that now has fallen off the DNA. So now we have no active repressor protein, and this gene is now turned on. Conversely, what happens to the RNA polymerase here when it stops at the repressor protein in the operator? Does it stay there and it's tied up forever for as long as the repressor protein is bound to the operator? Anyone have a guess there? Is it bound forever as long as the repressor protein is there, or does something happen? No one has a guess?
Does it just cut the RNA short?
And then what? Fall off? I'm trying to get you to say it falls off. The RNA polymerase is also binding to the DNA by a stoichiomic reaction, and every once in a while it'll fall off the DNA. So this RNA polymerase does not stay here forever. And the question was, does the RNA polymerase make a short RNA? I'm not sure if it does because RNA polymerase binds to the promoter and then it moves down the DNA and it will immediately get blocked at the operator. So I'm not sure it ever even makes an RNA. If it does, it'd be very short, just a few nucleotides. But it may not even make the nucleotides because it doesn't transcribe the the op the promoter, and it does transcribe the region of the operator, but it's not going to transcribe much, if any, of them. So if it makes an RNA, it'd be very, very short and only a few nucleotides. And it may not do anything. It may get blocked immediately at the operator, in which case it doesn't transcribe any RNA. But the point is the RNA polymerase is not going to be stuck here. It will eventually fall off, and then it'll bind to another promoter and then can transcribe that gene. And that makes sense because you don't want the cell to have a part of its machinery stuck and not able to do anything for the cell. All right, let me see if there's anything else I need to talk about. All right, so the sugar lactose is not around, at least for normal E. coli, unless it's in somebody's gut who's drinking milk or a dairy product or eating a dairy product. And so the lac operon is normally turned off in bacteria, but we do have a way of turning it on, and that's when allolactose is around. It'll bind to the repressive protein, which will free up eventually the operator, and then the RNA polymerase will transcribe the gene. And what's different is this lac operon will make 3 proteins, not 1. And that you can find in bacteria, but in eukaryotes, each operon only has 1 structural gene to them. So in a sense, they're a little more simple in that way, but in other ways, the eukaryote operon is more complex, and that is there's more more control of the gene. There's more regulatory genes, more regulatory units that control the gene expression. And that's all we're going to talk about eukaryote operons. All right, any question about controlling gene expression in bacteria? If not, let's talk about mutation. Mutation is a change in the genetic material. Here we're actually seeing a mutation, normal eye and a mutated eye. Mutations may be neutral, they may be beneficial, or they may be harmful. This mutation here is probably neutral. It probably is not going to affect the reproduction rate of the individual. But harmful mutation, can anyone mention a harmful mutation?
Sickle cell.
Yeah, sickle cell anemia. An individual born with 2 of those alleles will be unlikely to survive in adulthood. They live to about 20, and that might be with kind of medical care, and then they tend to die. I'm not sure if that's the case in first world countries, but they have health problems and they tend to die before they reproduce, actually, about age 20. There are also beneficial mutations. I once had or heard about a beneficial mutation, and that is there was a guy in his fly lab, and he was kind of lazy. And in the fly labs, you would have a cotton swab for the top of the cotton fly, and then you'd have agar or food in the bottom of the bottle, and then the flies would be in the bottle. And when the flies were pupating, they would crawl up, the larva would crawl up to the side of the glass and then form the pupa. This guy was kind of lazy and he never changed the food out until the food was so moldy that the flies would start starving and start dying in the bottle. And what happened was the flies would crawl up and they wouldn't find a place to form a pupa. There was no more room on the side of the glass for the fly to form a pupa. And what happened was he had in his lab a mutation happened where the fly could actually form the pupa on the cotton swab. That was the plug that you put in the bottle to, it's sort of the cap. And they would actually form their pupa on the cotton swab. And because of that mutation and this lazy guy who never changed the bottles, that mutation was selected for. So in that lab, that was a beneficial mutation. And it quickly became a very common mutation in the lab because the guy was lazy and he just never changed the labs and the bottles and the flies that had the old gene that had to do the pupa on the side of the glass, they were selected against because there was no place for them to form a pupa. Another beneficial mutation was about 10,000 years ago. Humans only had brown eyes. And in Northern Europe, we had the blue-eyed allele mutate from the brown-eyed allele. And initially, they think that gave the individuals a selective advantage. But now when they look at it, the blue-eyed gene is neutral to the brown-eyed gene, meaning neither has a selective advantage. And but they think the reason why the blue-eyed allele in Northern Europe became the most common, meaning the most frequent allele in Northern Europe, was they think that 10,000 years ago, for whatever reason, the blue-eyed allele had a selective advantage over the brown-eyed allele. And this is an actual case where before 10,000 years ago, humans only had one eye color, brownish. It could be dark brown, meaning essentially black, or regular brown or light brown, or it could even be greenish brown, but they were all brownish eyes. And that was from the brown-eyed allele being the only allele in the human population. But 10,000 years ago, we had a mutation, and the bluish-eyed allele came into being in Northern Europe. The blue-eyed allele now is beyond Northern Europe. In fact, the mutation happened in Australia, but independent of Europe. But and then the blue-eyed allele from people reproducing spread to other populations besides Northern Europe. Any questions about mutations? Nowadays, like I said, the blue-eyed allele is neutral to the brown-eyed allele, but they think initially it gave a selective advantage for individuals, and that's why the blue-eyed allele is the most common allele in Northern Europe. A mutagen is an agent that causes mutations. Something like cigarette smoke or phenol causes mutations in DNA. Now, you can have mutation without a mutagen, and that we call a spontaneous mutation, one that occurs in the absence of a mutagen. There are different types of mutations. There's a base substitution, also called a point mutation, and there are three types of base substitution. You can have a silent mutation, a missense mutation, and a nonsense mutation, and we'll talk about those probably in the next slide. Anyways, the base sense mutation is where one base is mutated, why it's called a point mutation. You can also have a frame shift mutation, and that is where one base is added or one base is subtracted from the DNA. And besides being a change in the nucleotide that's missing or added, the codons after that point all get changed, why it's called a frame shift mutation. A frame shift mutation can be more than one base added or subtracted. Like two bases can be added, and that's a frame shift mutation also. But if three bases are added, it's not a frame shift mutation, because if you add three bases, you're adding a another codon, so you're adding another amino acid, but all the other amino acids in the protein are the same. And that's why a frame shift mutation that happens to be 3 or a multiple of 3, either adding or subtracting, that's not a frame shift mutation. Is that clear to everyone? And the reason why it's not a frame shift mutation is the codons are not changed from the mutation. You do change that one codon, like if you add 3 bases, you're changing that one amino acid because it gets added to the protein, but all the other proteins are the same. So the codon frame shift is not changed. Any question about that? But if you just add one base, not only is that codon changed that gets the one base, but all other codons are changed because remember, codons are read by 3, and now we have 4. You simply take the first 3, and that's the first codon, and then that fourth codon becomes the first codon, the first, not codon, the first nucleotide of the next codon. And we'll take 3 codons again. You can see that'll change all the later codons. And that's why it's called the frame shift mutation. Any question about frame shift mutation? There's also another type of mutation called a macromutation. And this is not in the book, so you have to pay attention to the lecture for what a macromutation is. This is where an entire region of a chromosome is either added or deleted. And that's a macromutation. You should be careful of a macromutation because in genetics, a macromutation is used 2 ways, meaning a large region of DNA is changed, where an entire region of chromosome is either added or deleted. That is a macromutation. But a macromutation also means that you've changed a gene, which is a regulatory gene, and then that regulatory gene changes the expression of many other genes so that many phenotypes in the organism get changed. And that's not what I'm meaning by a mutation. I'm talking about where an entire region of DNA is either added or deleted. You actually know a macromutation where an entire chromosome is extra. And what do we call that? Where somebody is born with an entire extra chromosome.
Down syndrome.
Yeah, Down syndrome. I don't remember if it's chromosome 23 or chromosome 21. It's extra. I think it's chromosome 23, where the entire chromosome is extra. And that is a macromutation. Most macromutations in the human population are actually lethal, and then that will kill either the embryo or the fetus. Obviously, Down syndrome is an exception. Another exception is an individual born with only one X chromosome or 3 X chromosomes. Those individuals can survive. And then an individual born with XY chromosome. Those individuals appear normal, although they are chromosomal hermaphrodites, and depending on the expression of the genes, they could be a true hermaphrodite where one half of the body is female and the other half of the body is male. And there's also the XYY individuals, which appear to be normal, but in reality, they're not quite normal. And the XYY individuals tend to be higher in the prisons because it's thought that the XYY individuals are less likely to get along and more likely to cause trouble or crime, and so they're more likely to be in prison. But phenotypically and mentally, the XYY as well as the XXY individuals are phenotypically normal, and both of them can reproduce. The X individuals, which are actually called XO individuals, meaning they only have one X chromosome, they cannot reproduce. And I think the XX individuals cannot reproduce as well, but I'm not positive on that one. Anyways, those are some examples of macromutation. You will never find an individual with one human chromosome 1 or an individual born with 3 human chromosome number 1 because those are lethal and they will kill the embryo or the fetus. Any questions about a base substitution, a frame shift mutation, or a macromutation? All right, let's talk about a base substitution. That is where you change one nucleotide. Let me blow this up. That changes the DNA, and it may change the codon, changing the amino acid. So that should be a C there. It gets mutated and a T is put in there. When this T replicates, it will put in a TA across from that T, and now this mutation is fixed. The only way this mutation can go back is if the back mutation happens where that T is actually mutated to a C, and then it goes through a replication again, and then that will be replicated to a GC. Anyways, any question about a base substitution where you change one nucleotide? It may change the amino acid, depending on what the mutation is. We'll talk about that in just a minute. There are silent mutations. These are mutations where one nucleotide is changed, but one amino acid is not changed. This is because in the table of the codons, there are more than one codon for most of the amino acids. Like phenylalanine has 2 codons that code for the amino acid phenylalanine. And if UUU gets changed, mutated to UUC, the nucleotide is changed, but the protein is not, because UUC codes for the same amino acid as UUU. Any question about any of that? So a silent mutation does not change the protein. It does change the DNA, but it does not change the protein. And there's no effect on protein function. There's also a missense mutation, and that is where you change one base, and that results in a change in an amino acid. Let me see if I got this here. So we have the amino acid GGC that codes for the amino acid glycine, and that gets mutated to AGC. No, it gets mutated, sorry, to AGC. Yeah, that's right. And that will code for a different amino acid. Instead of glycine, it codes for serine. And that will be a missense mutation, where the nucleotide is changed, and then one base in the protein, meaning one amino acid in the protein, is changed. An example of a mutation like this is actually sickle cell anemia. I believe it's the 6th codon, meaning the 6th amino acid in the protein. Out of thousands of amino acids, one amino acid is changed, resulting in the sickle cell anemia. And that is a missense mutation. The blue-eyed gene actually is a missense mutation where you change the nucleotide and it changed the protein, resulting in the eye color being blue instead of brownish. There's also a nonsense mutation. This is where you change one nucleotide base, and that base results in a stop codon. So instead of putting in an amino acid, you code for protein translation to stop, and that will result in a truncated protein. Some muscular dystrophy and cystic fibrosis patients have a nonsense mutation, meaning they have a truncated protein. Oftentimes, the truncated protein will not function, but it does depend on how short the protein is and where the important part of the protein is. If the important part of the protein does get translated, the protein may function. It may not function as well as the normal protein, but it may function. But oftentimes, the nonsense mutation results in the protein not functioning. All right, let's talk a little bit more about frame shift mutation. That's where you insert or delete one or more bases, but not a multiple of three. Because if you add three bases, you only change that one amino acid. You don't change the coding reading frame after that change. Some Tay-Sachs patients are an example of a frame shift mutation. And here we have a frame shift mutation. Let me blow this up. Where we have AAA, and one nucleotide gets pulled out, A. So instead of having AAA coding for the amino acid phenylalanine, we have AA, and then it grabs the next nucleotide to read the codon, and it'll be AAC, which will code for the amino acid leucine. However, instead of having CCG for the next codon, because this codon gets used, we then begin the codon with CG and then take the next codon, A, and that will code for another amino acid, and the reading frame will be switched. So all other amino acids after this frame shift mutation may get changed. Any question about any of that? A frame shift mutation often is a severe mutation. It may result in non-functioning protein, and that's because all the amino acids after this frame shift will potentially be changed. You could have one that happens to be the same, like if A, and then there's a fourth A over here. If one of the A gets out, then it'll still be AAA. But generally, most of the amino acids will be changed, and potentially all of the amino acids could be changed. All right, let's talk about the causes of mutation. There are two causes of mutation. There can be a spontaneous mutation, and this just happens normally when the DNA is being replicated, mistakes get made, and that's a spontaneous mutation. And then you can have a mutagen causing the mutation. There's two types of mutagens, radiation and chemical mutagens. Radiation, there's two types of radiation, ionizing radiation, like X-rays and gamma rays. These have a lot of energy in them. And when that energy hits an electron, that electron absorbs the energy, giving that electron so much energy that this electron actually will leave the atom. This ion can then bump into DNA and then mutate the DNA. Because this ion will actually want to gain an electron, and whatever it bumps into, it tends to steal an electron from it. If it bumps into DNA, that can mutate the DNA. We're not going to talk about repair mutations. The second form of of radiation mutation caused by radiation is a mutation caused by UV radiation. Let me blow this slide up. When UV radiation hits DNA, it will damage two thymines which are next to each other. What will happen is the UV radiation will hit the thymines, causing a thymine dimer to form between the two thymines, where the thymines actually have two covalent chemical bonds between them, why it's called a thymine dimer. This will create a bubble in the DNA, or a, well, let's just call it a bubble, where the thymines will be bound together. They're not normally. And then these thymines will not hydrogen bond with their corresponding A's in the other strand of DNA. This region of the DNA will suffer DNA DNA replication will not happen past this thymine dimer, and then RNA transcription will not happen past this thymine dimer. Any questions about thymine dimers? Like I said, we're not going to talk about repair. All right, let's talk about a chemical mutagen. We're going to talk about nitrous acid as a chemical mutagen, and this is the only chemical we're going to talk about as a chemical mutagen. Nitrous acid will come to the DNA and then change the DNA, where this A will be chemically changed. It's still an A, but it doesn't look like an A to the cell. Instead of looking like an A, it will actually look like a G, so that when the RNA, not the RNA, the DNA polymerase is replicating this batch of DNA, the nucleotides, it'll see the C and put a G, it'll see the G and put a C. It will see this changed A, but not recognize it as an A and recognize it as a G, and then it'll put a C corresponding to this changed A. And that is how nitrous acid mutates the DNA, changing the A so that it's no longer read as an A, it's read as a G, and then the C is put opposite it. And then when this replicates, instead of getting the AT, which should be in this third base position, we're going to get a GC in the base position. Any question about how nitrous acid acts as a mutagen? All right, let's talk a little bit further about the frequency of mutation. A spontaneous mutation happens all the time when DNA is being replicated, but it happens about one time in 10 to the 9th replicated bases, meaning when DNA polymerase is replicating the DNA nucleotides, it'll have to replicate 1 times 10 to the 9th bases before it will make a mistake and add the wrong base. Because a gene has about 1,000 on average nucleotides for the gene, that means the frequency of mutation per gene is 1 times 10 to the 6th, meaning there will be 1 mutation in 1,000,000 individuals or organisms. And that's the spontaneous mutation rate. 1 times 10 to the 9th replicated bases, or if we're talking about genes, 1 in 1,000,000. 1 in 1,000,000 isn't a very large number, but if you're talking about something with as large as the human population, which has very close to 8 billion, or maybe it's even now 8 billion, I don't know, people, That means every year in the world, we are having children who are born with mutations. Because about 1 in a million children being born will be mutated for one gene of interest. Any question about any of that? All right, mutagens increase the mutation rate to something like 10 to 1,000-fold. So instead of being 1 in 10 to the 9th replicated base, which will be mutated, it'll be 1 in 10 to the 8th bases at a minimum, because we're saying a mutation increases the mutation rate between 10,000 and 100,000. That means that the mutation rate per replicated gene will be 1 in 10 to the 5th genes that will be mutated to 1 in 1,000 genes if its mutation rate's 1,000 times greater. Any question around that? All right, identifying mutants experimentally. You can test for mutants by positive direct selection or by negative indirect selection. Positive direct selection detects mutant cells because they grow or appear different. Like if somebody were to see that walking down the street in Washington or Oregon, you would go and say, yep, there goes a mutant, right? Positive direct selection. You can see the individual as a mutant, and so you identify it that way. There's also negative indirect selection, and this detects the mutant cells because they do not grow. And that's because they're a mutant. You can test negative selection via replica plating. I used to do this when I was an undergraduate student, and that is you get a plate having colonies separated like this, and then you make a velvet stamp of the plate. When you push the velvet into the plate, the cells from the colonies will be put on the velvet. And then you stamp the velvet on two different plates, one having media containing the amino acid histidine, another having media lacking the amino acid histidine. Oops, sorry about that. You then incubate the plates and you look for a plate which has something growing from, what is it, this cell there maybe, this colony here, where it will grow on the plate containing the amino acid histidine, but it does not grow on the plate lacking the amino acid histidine. So this mutant is a mutant in the ability to make the amino acid histidine, and if you don't supply the amino acid histidine, these cells will not grow because they're a mutant. Anyways, this is an example of negative selection, where you're looking at the fact that these cells here, I think it's there, will not grow because they're mutant if you do not supply the amino acid histidine. If you do supply the amino acid histidine, they will grow. Any question about any of that? All right, any question about identifying mutants? If not, let's move on to genetic recombination. Recombination is the exchange of genes between two DNA molecules. Crossing over is when two DNA molecules come together, like the chromosome A of the mother and chromosome B of the father, and then they come together and they cross over, literally, like that, and then they break right here, where the mother's chromosome combines with the father's chromosome. So this will be the mother's chromosome here, and the second part of the chromosome will be the father's chromosome. What do you notice that's different between crossing over and recombination? Is crossing over always recombination? Or is recombination always crossing over? Anyone? All right, crossing over is always recombination. Recombination is frequently crossing over, but it is not always crossing over. Recombination is a little more general term where two DNA molecules exchange parts. The crossing over is something that happens in mitosis and meiosis all the time, where the mother's chromosome comes together with the father's chromosome, and then they cross over, the chromosome breaks, and then the chromosome is a new chromosome having portions of the mother's chromosome and portions of the father's chromosome. Any question about genetic recombination? If not, let's move on to genetic transfer. We talked about vertical gene transfer occurs during reproduction or between generations of cells when the parent cell has its DNA flow to the offspring cell or the daughter cell. That's vertical gene transfer. Horizontal gene transfer is the transfer of genes between cells of the same generation, which is the same as saying that DNA comes from a donor cell and it moves into a recipient cell. That's horizontal gene transfer. Horizontal gene transfer in bacteria can happen in three ways. It can happen by transformation, by conjugation, and transduction. Transformation is where DNA comes out of a cell, exists as a naked piece of DNA, which is then brought into a recipient cell. Let me blow this up. And then at least part of that new piece of DNA are added to the chromosome. And so we'll get the little A gene added to our chromosome. And now this cell has gotten new DNA from this DNA, and it will eventually recombine. And then that little A, which came from this DNA outside the cell, will be expressed because little a is put into the chromosome. It'll be expressed and it could change the phenotype of this cell. This is a genetically transformed cell because a gene that it lacked initially, the little a gene, came from this piece of DNA. Suppose little a was a gene encoding antibiotic resistance. Would that be a concern for this bacteria? Assuming this bacteria were to cause an infection in a patient? Anyone? It's either a yes or no question once again. All right. Well, if no one's going to ask, I'm just going to leave it at that. Here's a picture of transformation where a cell sees a piece of DNA, consumes it, gets indigestion when the DNA is beginning to be transcribed and translated, and then it could change the phenotype of this cell, as shown here. And that's from the gene or genes from this piece of DNA there. Coding for something new, changing the phenotype of this cell. Any questions about transformation? All right. Gene transfer can also happen by conjugation. This is where DNA moves from one cell into another cell. conjugation happens in gram-negatives with the sex pilus. Let me blow this slide up. So gram-negatives from this cell here has an F plasmid, and it makes a sex pilus. The F plasmid actually codes for the sex pilus to be made. That sex pilus can come up to a F minus cell, one that's lacking the plasmid, lacking the sex pilus, and literally skewer the cell. And then that sex pilus can actually winch the two cells together, although it doesn't need to winch them together because the sex pilus is hollow. This cell here can then replicate its F plasmid, making two copies, and then send one plasmid into the F minus cell through the sex pilus. Remember, the sex pilus is hollow, hollow. And that actually connects the cytoplasm of this cell to that cell. But the cells do not need to be this close together. They could be far apart, as shown in this picture here, fairly far apart. And that's because the plasmid can move across the hollow sex pilus. Once this plasmid then moves into this F minus cell, that F minus cell changes to an F plus cell because it has the F plasmid in it, and the F plasmid will code for this cell to make its own sex pilus. So now we have two F plus cells, both of which have an F plasmid, and both of which will have a sex pilus. And that's changing the F minus cell to an F plus cell. Any question about any of that? Anyways, that's called sexual conjugation. If you are confused on why it's called sexual conjugation, you can ask your significant other. That's a joke, by the way. All right. Sexual conjugation in gram-positives does not happen with the sex pilus. Sexual conjugation happens in gram-positives with the mating bridge. The two gram-positive cells come together, and then the cell wall dissipates, and then the cell membrane of this cell confused with the cell membrane of that cell. So the cell membrane then comes around the cell this way, comes over here, comes here, and then fuses to the cell membrane there. So the cell membrane comes around and then there. Any questions about any of that? That literally means that the cytoplasm of this cell is connected to the cytoplasm of that cell. And then a plasmid from this cell can move from this cell, has to replicate, move from this cell, and then move into that cell, or vice versa. A plasmid from this cell can replicate and then move into that cell. And that's how a mating bridge happens in a gram-positive cell, where the two cells come together, their cell membranes fuse, connecting the cytoplasm of the two cells together. The difference is that in the gram-positives, in the mating bridge, there is no plasmid specifically in one of the cells that moves into the other. It could be any plasmid from this cell moving into that cell, or any plasmid from this cell moving into that cell, or vice versa. With sexual conjugation in a gram-negative, you always have the F plasmid replicating in this cell and then moving through the sex pilus into the F minus cell. Any questions about sexual conjugation? All right. I have a few more slides here, but I'm going to stop here because we have a lab at 6:30. I'm going to start the lab at 6:32, so you guys will have a 10-minute break. I didn't notice that the uh, the time has expired. Any questions?
Yeah, I have a question. When are labs going to be regraded?
I'm going to talk about two labs today in the lab that have been regraded.
Okay, so they're all going to be regraded eventually, though.
They will all be regraded a second time. I don't think any of
Okay.
them have been graded a second time yet, but they all will be graded a second time.
Okay, and also, um, for the unknowns project, for the gram stain, do I do I also look at the arrangement of it or no? Just the shape and the positive or negative?
Okay, why don't you ask that in the lab because that's actually a lab question.
Okay, sorry.
It's not a problem, but I do kind of want to end it here. But
Okay.
like I said, that is a lab question, so why don't you ask that one in the lab? And
Okay, thank you.
I'll ask that at the start, answer that at the start of the lab.
Okay, thank
Okay?
you.
All right.